Hello, everyone, and welcome to today's CryoEM Current Practices webinar. You're joining the 3 national centers established by the NIH Common Fund Transformative High-Resolution Cryo-Electron Microscopy Program. My name is Christina Zimani. I'm a scientist and training liaison at the National Center for CryoEM Access and Training, NCCAT. With me today are my colleagues at NCCAT, Ed Eng and Eugene Chua. Joining us from the Stanford SLAC CryoEM Center S2C2 is Michael Schmidt, and from the Pacific Northwest CryoEM Center PNCC is Lauren Vega. This webinar is a regular event that we host to highlight the methods and tips and tricks that groups are using to collect and interpret the data that they can collect using National CryoEM Center resources. We typically meet the last Thursday of every month, but because of the concentration of holidays, we've shifted our schedule for the end of the year, and our last 2022 date will be December 15th. And actually, in place of a formal talk, we're going to host a roundtable discussion with a panel of 3 or 4 center users focused on the topic of particle picking using neural network pickers. So our panelists will share some of their practical experience with this part of the cryo-EM processing workflow, and we'll have an open forum for Q&A. This will be sort of in meeting format instead of webinar format, so you can ask your questions in person to some more experienced practitioners. And a little spoiler, we may even have some of the developers of these software tools themselves pop in and join us. So look out for our announcements and registration link for that date coming soon to your email, or you can check on the cryoemcenters.org website for registration link for December 15th. For today, Eugene, who is the champion of the grid preparation and screening access at NCCAT, will introduce our speaker, Robert Monsen, after I give each of my colleagues a chance to give a quick update from their respective centers on the resources we make available at no cost to the research community. We are recording today's talk, and you'll be able to find this recording along with all of our past talks on our YouTube page or through the events page at cryoemcenters.org. And for final Zoom logistics, we're going to be watching the Q&A box for questions. So please use the Q&A box and not the chat to send in your questions, and I'll be moderating those at the end of the talk. So with that, I'm going to turn over to our center representatives, and Lauren, why don't you go first?
Thanks, Christina. So for PNCC, I am the project coordinator, and at PNCC, we offer 2 proposal types to get access to our center. So we have a screening access proposal that is up to 5 days of on-site sample preparation optimization that is targeted to low resource institutions. We also have our general access that's for both single particle analysis and tomography and gives up to 480 hours a year and is valid for 2 years. So those approved general access proposals are eligible for our training workshops. We kind of filter you in through our training workshops to provide training. And we also have a monthly submission deadline for all of our applications. So each approved application that comes through at PNCC is delegated what we called a scientific point of contact, or SPOC, to ask your scientific questions. We have 5 scopes. So we have an Arctica with K3, and we have 4 Krioses with K3. 3 of those have Falcon 3, and 1 has been upgraded to Falcon 4. 3 of them are also equipped with energy filters and have been upgraded to fringe-free imaging, with the final one undergoing that this month. For our sample preparation, we have our Richer bot and Leica GP2, but our Richer jet is back at the factory. Last month, we ran our sample preparation workshop, and we'll be holding a data processing workshop in November and our CryoEM introductory training workshops in December, both of which are remote. So apply today if you're interested.
Thank you, Lauren. And Mike, I'll turn over to you now.
Thanks, Christina. I'm Mike Schmidt, and I'm one of the co-directors of the S2C2 CryoEM Center at Stanford SLAC. We have 3 Krioses, one of which has newly installed cold phag. They're equipped with K3 or Falcon 4 cameras. We have a Talos Arctica and a newly, well, to be installed Tundra, which is coming on, and and another Krios, which will make our full set of 4 Krioses available by the end of the year, or maybe early January. We run both high resolution data collection projects and screening projects and training projects, all of which have multiple users coming on site now. We're welcoming people to come on site or send your samples remotely by shipping them. We have remote access and remote microscope control for people that want to stay off site, and we welcome all types of users from all sorts of institutions all over the country. Please join us. There's our website. We can be contacted through that or the cryoemcenters.org website. Thank you.
Thank you, Mike. And now I'll turn over to Ed.
Hi, my name is Ed, the manager of NCCAT here in New York City. We offer 3 main access types. One is high resolution data collection on our 4 dedicated Krios instruments. We offer a grid preparation and screening service, which you'll hear a little bit more about, for access to our plunge freezers, chameleon blot-free vitrification device, as well as a Glacios, and a robust cross-training program that has access to a Tundra that's going to get signed off this quarter, I hope. In addition, we do allow on-site access. And if you are going to come to New York City for a long period of time, ask us about subsidized housing. Something that we're doing every year is a short course. This year we had a single particle short course. Next spring we'll have a tomography short course, and applications are open for that TOMO short course in April. If you want to find out more, go to nccat.nysbc.org, and now I'll turn it over to Eugene to introduce our speaker.
Right, thanks, Ed. It's my great pleasure today to introduce Rob. Rob is a postdoc at the University of Louisville, where he studies G-quadruplexes. He uses an integrative structural biology and biophysical approach to study these G-quadruplexes. And Rob is an expert in small angle X-ray scattering, molecular dynamics, and hydrodynamics methods to study these quadruplexes. And now also, I think, a cryo-EM expert. Rob was our inaugural participant in our grid preparation and screening program and now we intake approximately 1 user every 2 weeks for a 3-day on-site visit, which was what Rob started with. So I think today he'll be sharing with us a little bit about the work that he has done here and also that he does at his lab. So take it away, Rob.
All right, thank you, Eugene, and thank you everybody for having me. Actually, I need to share my slides real quick. Oops, where's the share? Okay, can you see that?
Yes, we can now. Thank you.
Okay, sorry about that. Yeah, so I'm Rob Monsen. I'm a postdoctoral researcher in the lab of John Trent at U of L Brown Cancer Center. And I'm going to be talking about a DNA G-quadruplex system that we've been trying to characterize for a while, and how cryo-EM has helped us sort of resolve this, a misconception that's been in the field for quite some time. So first, what are G-quadruplexes? They are 4-stranded DNA structures composed of, let me see, can you see its pointer?
I can, yes.
Okay. They're 4-stranded DNA structures composed of stacks of guanine tetrads, and a G-tetrad is shown here on the left. So it's 4 cyclically bonded guanines, and they form the square planar kind of shape. And so we often represent them schematically as just squares. In the central channel here, you see a cation. So typically sodium or potassium are within these channels, and these help support the structure by neutralizing the partial negative charge from the inward facing carbonyls. So in B here, you can see just a schematic, a ribbon diagram of a parallel G-quadruplex. And then below here, you see 3 different conformations, schematics of different conformations of G-quadruplexes, with the green and arrows showing the sugar phosphate backbone and its directionality from 5 to 3'. So if you were going to search for one of these quadruplexes in the genome, the traditional motif is shown down here. You have 4 guanine tracts separated by 3 loop regions where the guanine tract X here is between 2 and 4, and Y can be highly variable, but at least 1. So where are G-quadruplexes in the genome? Traditionally, we associate quadruplexes with the free 3' overhang of our telomere, the ends of our chromosomes. And this region has these hexanucleotide repeats, TTA, GGG, which enable the formation of G-quadruplexes and actually can form higher order assemblies of G-quadruplexes. So the quadruplexes here play a role in telomeric homeostasis. And it turns out if you stabilize them, can actually inhibit telomerase activity. So for a long time, quadruplex research has focused on this region to find small molecule stabilizers for cancer treatments. But more recently, bioinformatic studies as well as chip sequencing studies have revealed that G-quadruplexes are also non-randomly distributed in the promoters of many genes. Actually, many highly transcribed genes is what's been shown most recently. And so it turns out, and we're still working out the mechanisms of how this works for a lot of promoters, but for the most part, what we know is that G-quadruplexes can interfere with RNA polymerase activity as well as inhibit or recruit transcription factor binding. And so stabilizing these structures can actually modulate promoter activity. And we're really interested in promoters specifically because of we can actually, in some cases, downregulate difficult to target proteins, such as the case of c-Myc. So this is a 2002 paper by Siddiqui, Jain et al. And PNAS, and it's kind of a seminal paper in the field. The figure here is just showing a model from a study in the c-Myc promoter. c-Myc is a notorious oncogene that's upregulated in 70 to 90% of all cancers. So what they did was they found a G-quadruplex in its promoter and found that it's a highly stable quadruplex that negatively regulates gene transcription, basally. And it requires destabilization by proteins to enable its transcription to turn on. What they did in this study is took this very high affinity G-quadruplex ligand called TMPYP4, bound it to this quadruplex and showed that you can lock this promoter into a transcriptional off state by stabilizing the quadruplex. So this kind of gets at the heart of what we're trying to study these promoter quadruplexes for, which is, drug discovery efforts. But another reason why I'm showing this schematic here is because although at the time this was very innocuous, drawing the schematic like this, it has actually become sort of pervasive in the field. And so this gets back to the sort of title that I have going for this talk is Oxford Dictionary's definition of a meme is an idea that is passed from one member of society to another, not in the genes, but often by people just copying it. And so in 2002, this was the first sort of quadruplex in a duplex context schematic rendering in the literature. And it turns out that as of 2 decades later, we're still drawing it the same way in all of the publications when we're showing our different schematics and models for our systems. But really, there's no physical basis for us to draw these quadruplexes like this. And in fact, intuitively, it doesn't make sense because G-tetrads in general are very hydrophobic. So we would expect that they would be more sequestered and away from being so solvent exposed. And actually, in putting together this talk, there is another ongoing seminar series for the G-quadruplex community that are also using this same kind of duplex quadruplex schematic as shown here. They have the quadruplex popped out and just kind of hanging out, but really we don't know if that's actually the case. And in our opinion, it doesn't seem like it should be the case. And so why hasn't anybody actually characterized a promoter G-quadruplex bubble before? Well, long flexible DNA are traditionally not amenable to high resolution structural determination methods like NMR and X-ray diffraction. There's no natural, well-behaved promoter G4 model system, so you can't just, cut a G-quadruplex promoter out of a genomic environment, put it into a test tube, and investigate the G-quadruplex formation and its properties. This is largely due to the fact that when G-quadruplexes are in the presence of their complement, they readily undergo conversion to a duplex. So inherently, they're very difficult to handle in vitro. And then G-quadruplexes themselves are very sensitive to their environment. So they're unstable if you remove the sodium and potassium cations that are in the central channel, such as if you were to do a significant amount of washing steps with water. And then they can also change topology if they're in a chemically crowded or packed into crystals kind of environments. So this is another reason why X-ray diffraction is not always applicable to studying these systems. So actually, one of the first studies that tried to visualize G-quadruplex in the context of a duplex, so in the most biologically relevant situation as possible, is this Duquette and colleagues in 2004. So what they did was they wanted to get electron micrographs of a G-quadruplex system that they inserted into a plasmid. And so what they did pretty cleverly was they did in vitro transcription of the complement strand to form what is a hybrid RNA DNA or R loop. And so by doing this, you can sequester that complement strand and displace the G-rich strand such that it can form quadruplexes and you might be able to visualize them. However, this is the micrograph, or the main micrograph from that study, and you can see that there is a bubble where they have done in vitro transcription. They formed the R loop and displaced what in one case is the G loop, but really the resolution is so poor that they couldn't actually, there's nothing actionable here. There's no way to build a model against this or anything. So and it's also up to debate if they even see quadruplex formation there. So about 5 years later, Neves and colleagues published a study doing a similar thing. So this is by atomic force microscopy. And they used a plasmid as well with G-rich inserts to form quadruplexes. And using the same in vitro transcription method, they were able to capture out similarly these loops, as shown here in A, B, and C. And the authors themselves say that the shorter region, which is the thicker, denser region here, corresponds to the G-quadruplex region. And then the part that's looped out, the RNA DNA duplex, or R loop. And they've drawn schematics as well to show how they interpret this data. And so you can see that in each case, the quadruplexes really aren't just sticking out. I mean, here sort of, but realistically, what they're seeing is the R loop protruding away, which is kind of in contrast to what the original schematic renderings would lead you to believe. And so this brought us all to this one simple question that we wanted to ask. So what does a promoter G4 look like in the context of a duplex? So does it look like the schematic renderings where you have a quadruplex popped out and fully solvent exposed and accessible with the complement strand just hanging out on the other side? Or is it more occluded, where the quadruplex might be interacting at its kind of hydrophobic faces with the rest of the DNA? So these are the two models we were trying to investigate. So in order to do this, we created a synthetic duplex quadruplex duplex model, which we call DGD. And just for reference, it's only 28.5 kilodaltons, so it's very small relative to what you do in a cryo-EM experiment typically. But what we did was we took a very well-characterized quadruplex called PDB1XAV. This was solved by NMR, solution NMR, and it is a parallel conformation G-quadruplex, which we've found and others have shown is the most common promoter conformation of a quadruplex. It's also a model system for quadruplex studies because it has a very high thermodynamic stability. So we were able to kind of stack the deck and ensure that we get an actual quadruplex to form. And then we also designed artificial kind of arbitrary duplex arms on either side to mitigate any competing structures from forming. And then opposite of the G-rich insert, we have a poly-DT loop And so this has the same number of nucleotides as the G-rich insert, so that we can maintain the same length of sugar phosphate backbone, but we have thymine here, which will prevent interaction with the quadruplex to stop it from forming or something. So the first thing that we did was we ensured that we're getting quadruplex formation and the complements are binding on either side without disrupting quadruplex stability. So the first thing we did was analytical ultracentrifugation studies. So what this shows is, so the G strand alone sediments at about 2.5. And when we mix in this pseudo complement with a poly-DT region, we get a doubling of molecular weight and shift in sedimentation coefficient consistent with the duplex occurring. And then we also looked at the proton NMR spectra. So if you look between about 10 and 14 parts per million, these are fingerprint regions for Watson and Crick proton shifts, which we see. So this confirms duplex formation. And then we also have Hoogsteen hydrogen bond shifts. And these are characteristic, this region is characteristic of G-quadruplex iminoproton shifts. And we have approximately 12 partially overlapped peaks here, which confirm that we have our 3-stacked G-quadruplex. So this is all good confirmation that we have what we want to see. As Eugene said, we've also been doing a lot of small angle X-ray scattering studies. And so this has been in collaboration at Argonne National Laboratory in Chicago with Drs. Chakravarthy and Hopkins. So in the top left here is the buffer subtracted scattering for the DGD system. And you can see it's plotted as intensity versus momentum transfer, or Q. This can be thought about as the scattering angle. And what we can do with this data is we can turn it into a real space histogram or weighted histogram of the interatomic distances of our particle. This is called a P of R distribution using an indirect Fourier transform. And what this gives us is general shape information of our particle overall, the inner particle distances, but it also gives us an estimate of our maximum length of our particle. And this is important for, of course, for particle picking if you have sort of an unknown that you're looking for in your micrograph. But another cool thing that we can do with this P of R distribution data is you can use it in ab initio modeling tools to try to generate a solution, not a unique solution necessarily, but a solution to your data to get a 3D representation of the P of R distribution. And that's shown here. And what you can see is pretty readily is that there is what looks to be a duplex axis across the bottom here. And then there's a central density that protrudes only in one direction, but it's not a cruciform. So this is kind of our first indication that what we're getting is not quadruplex and reverse complement sticking opposite of each other, but instead what it's more consistent with is the quadruplex sitting in between the two duplex arms with the poly DT doing something. I almost hesitate to show this data just because the resolution here, theoretically is up to maybe 20 angstrom resolution, but realistically the estimate on this ab initio reconstruction is like 37 angstrom. And again, it's not a unique solution. So it's very difficult to really say one way or another based on this data alone, how the quadruplex is situated amongst the rest of the construct. So it was around this time when I came across this paper from Huachu's lab at the Stanford University, the SLAC, where they were looking at an RNA of a 40 kilodalton SAM4 riboswitch RNA. And they actually were able to produce a map of nominally 3.7 angstrom resolution, and it's shown down here. And so I thought, this is pretty amazing, really impressive stuff. And you can even see, the tertiary overall folds, as well as in some cases they showed base pair resolution, which I was impressed by. And so, of course, we don't have any cryo-EM facilities at University of Louisville in Kentucky, but I reached out and found that the NCCAT group, they at the time had general user proposals that I could apply for and try to do a grid preparation and screening and all that. And I even did a grid making course when I went out there. But yeah, this is me and Ed. And so I took my samples out, and Eugene kind of showed me the ropes of cryo-EM preparation and screening and analysis. So on the left here is the original vitrification conditions and screening conditions. So our samples we keep in typically just a standard sodium phosphate buffer with 185 millimolar potassium chloride. And this amount of potassium chloride is necessary to maintain the integrity of the G-quadruplex. And then we also have 1 millimolar EDTA, which prevents DNAs. So very simple buffer. In all cases, the vitrification was done by Vitribot Mark IV with 100% humidity and at 5 degrees C. All of the grids were glow discharged in an argon oxygen environment. And we initially used quantifoil grids, both copper carbon and carbon copper and carbon gold grids. And we started with a concentration of 0.5 mg per mil, which is about what they used in the RNA paper. And then various blot times, at least initially, to try to get an ideal ice thickness. So this is one of the first micrographs that came from the Glacios screening on quantifoil grids at 0.5 mg per mil. And you can see that, well, you probably can't see because the particles are so small. They're very difficult to see. There's probably, one for sure right there. But really, it was looking pretty bad at first. And we also had also my first time clipping and everything, so a lot of mangled grids as well, which were my fault. But during this whole process of Glacios screening and imaging and trying different concentrations, we found that realistically all I needed to do was actually clip properly and increase the concentration to about 10x, 5 to 10x what we were initially using. And we were going to use quantifoil grids, but actually during this whole week of the grid making course and prepping all these grids and screening them, Alex Way actually gave us a few of his secret ultrafoil gold grids, which I believe were pretty scarce at the time because of COVID. But we ended up using ultrafoil gold grids at 5x concentration and 4-second blot time. And so you can see this is a Glacios image collected on the ultrafoil grids, and you can see all the little tiny black specks everywhere that are our particle. And so the left is just showing our final vitrification and sample conditions. And on the right, just showing from the Glacios, we could actually measure some of the longest distances of these particles, which showed good agreement with the Dmax estimate that we had from our small angle X-ray scattering experiment. So this was really reassuring. So data collection was done on a Cryos operated 300 kV with Falcon 4 EC imaging system. In total, 12,175 movies were collected. The right here is a representative micrograph from the Cryos. And then below it are some initial 2D class averages using the max dimension from SACS in our elliptical blob picking. And you can see that in general, the overall kind of size and shape is consistent with what we expect and sort of like what we see from the SACS ab initio, but we do see kind of a distinct bend angle in each case with a middle central density. So this is the full CryoSpark workflow that I used, and I'm more than happy to stop here or come back to this if there's any questions. So we started with 12,000 micrographs initially. These were curated down to 9,045 by CTF fit resolution, ice thickness, astigmatism, and defocus. I did blob picking, 2D classification, 3D ab initio, and then used the best reconstruction from here to do template picking. From the 2D classifications of that output, there were about 666,000 particles in what seemed to be the good classes. These were then used in a 3D ab initio with 3 classes. Heterogeneous refinement, I removed the junk particles from that, which resulted in 303,000 final particles, and these all seemingly were good. So I initially used these to build a final reconstruction using non-uniform refinement, but it really was featureless. And so I suspected that there may be something else going on, such as flexibility. Eugene actually suggested trying 3D variability analysis in CryoSpark, and it turns out that it did seem like there was some significant amount of flexibility among these particles, or at least different kind of conformational states that were very similar. So from there, I did a 3D ab initio using this particle stack, 3 classes, and empirically found that 3 classes and 0.8 similarity gave the best results. So there were 3 classes that were kind of distinct in their bend angle and lengths. And class 1 here was the most readily interpretable. As you can see, there is a right-handed B-form kind of duplex looking grooves on either side of this thicker central density. So for the modeling purposes going forward, this is really the one class that I'm going to stick with. The most compelling part of the cryospark analysis, in our opinion, is shown here. So the bottom are the non-uniform, refined, and sharpened maps at 2 different thresholds for each of 3 different classes, class 0, 1, and 2, from that ab initio refinement. But what I thought was most compelling are these 2-dimensional, or these heat map slices from the non-uniform refinements. And because you can see clearly there are 2 duplex handles as expected on either side of a very globular, very central density here, which we take to be the quadruplex because we know it's stable and very globular. And then also you can kind of see what's kind of a washed out looking density that we attribute to probably the poly DT region, which makes sense from the SACS data that it would be kind of sticking and protruding in one direction away from the axis of the helix. So this was, you know, really kind of confirming that the quadruplex is sitting between these 2 arms. However, there is a distinct bend angle and almost a continuous distribution of unstacking from one side, which is really surprising. And so, yeah, so down here is the cryo-1 refined map, and you can see very well defined right-handed B-form duplexes on either side. The central density where we expect the quadruplex is actually has sort of a flat surface on one side, which is consistent with the G-tetrad face. And then we have something going on here on the top with missing density in the middle, which appears to be where the poly DT loop would be sitting. And I thought that I would, it's necessary I show this. So this is the orientational distribution from CryoSpark, showing that this, aside from a few angles, we have mostly good coverage in most viewing directions of the particles. And then on D here is the gold standard FSC resolution map, plot showing a nominal resolution of about 7.4 angstrom. And then down here is the local resolution estimates, with the blue region corresponding to the lowest confidence or lowest resolution region, which again, we believe this region corresponds to that poly DT loop that could have a lot of flexibility. So in thinking about how we could model into this cryo-EM map, and as well as when we were trying to model against just the SACS data alone, and because our lab is a modeling lab, I constructed this model with a duplex arm here, the quadruplex in red, a duplex on the other side stacked, and then built in the poly DT loop. And what we found from initial simulations is that the 3' end on the right here immediately becomes unstacked and actually starts to exhibit conformations or bends across this axis that are very similar to what we were seeing in the cryo-EM maps. Which is, coincidence. There's no bias here. This is just kind of what happened. And so we took advantage of these bend angles because they were fitting so well with the cryo-EM data that we were able to do, we were able to fit the initial bent conformation that best fit from the original MD that was unrestrained, fit it into the map manually, and then start a simulation using what's called self-guided Langevin dynamics with EMAP, which essentially is just a flexible molecular dynamic simulation with the map being a constraint so that you can flexibly fit this poly DT loop and the quadruplex and duplex can relax with the restraints of the map being there. And so you can see how it's kind of fitting in throughout the course of the simulation. The final map correlation value was 0.93 here. And so this is just kind of spinning it around and showing the different views. So the duplex and quadruplex regions in our minds fit very well to the data. 98% of all heavy atoms are within the map. And you can see that the poly-DT loop in yellow here, it does fit very well inside this density, but we were also kind of worried that if we were missing some density in this map, that it could be kind of smushing the poly-DT loop in there based on our MD refinement. And so at this stage, I conducted much more extensive MD simulations, starting from the refined coordinates that we have from the cryo-EM refinement. And so what I did was carry out duplicate independent 500 nanosecond, so total of 1 microsecond explicit solvent MD simulation, starting from those initial coordinates to assess by MD if this system is even stable at all. And if so, what is it doing in solution? And so what I found was sort of surprising in that, so the G-quadruplexes, these are showing heavy atom RMSDs across the trajectories. So for the G4, which is the central here, the central part here, the heavy atom RMSD plots show less than 3 angstrom deviation, which suggests that for the most part, it's essentially unchanged throughout all the simulation. However, the poly-DT loop initially in both cases has a very quick relaxation time. And what it does is it actually, some of the poly-DT residues actually preferentially stack at the 3' G-tetrad interface here and actually remain stably stacked there throughout all the simulation. And so there is relatively little movement of the poly-DT loop aside from some nucleotides out here. And then the G-rich strand, so this is essentially just the quadruplex strand spanning from end to end across this axis. And it turns out that the bulk of the movement is actually coming from this bending and twisting across this full axis here. And you can see I've got this kind of showing it. There's a bend and a twist that goes on here that in reality is fairly consistent with the class 0 and class 1 heat maps that we saw in the cryo-EM non-uniform refinements. And so to us, this makes a lot of sense, but also shows that both interfaces of the G-quadruplexes are actually very occluded. That's, oh, I'm sorry, and I need to admit, so this is the first principal component of movement from the first MD simulation, one of the 2 MD simulations done here. So this isn't an actual MD simulation, this is just the first component of movement showing the major fluctuation that's going on throughout these simulations. So it is fairly stable interacting here. So lastly, we're not doing this just as an academic, I told you so. We're interested as a drug discovery lab in finding better small molecules that target specifically G-quadruplexes. So, I mean, only 2 or 3, a small handful of G-quadruplex binding ligands have actually made it into clinical trials. And that's not through clinical trials, that's into. So clearly we've not done a very great job as a field in identifying specific drug-like small molecules. So, and so what we were doing with this is what's called a sitemap druggability analysis. So what we're doing here is comparing these 2 models. So this is the DGD model with the poly-DT swapped for the actual reverse complement. And this is the 1XAV quadruplex that we use that in our DGD model. This is derived from the NMR structure. I'm sorry, this is the NMR structure. And so what sitemap does is it takes in these 2 models and it searches the entire structure and identifies the best, most druggable sites in both cases. And using a scoring system that's been empirically determined using high and low affinity protein ligand interaction systems, it assesses each of it assesses each of its sites and scores it based on the size of the pocket, degree of enclosure, and subtracts for any amount of hydrophilicness. So a more hydrophobic deep pocket would be scored better than something very hydrophilic. And what you can see here in purple are the 2 sites identified. And what you see for DGD, there's a very nice big groove here between the duplex axis and the interface of the 5' end of the G-quadruplex, as well as it creeps up into the loop of the quadruplex, as well as partially this reverse complement region. Whereas with a small single quadruplex alone, there's not a lot of real estate to target. And so you see this little pink, wad of gum here. There's just not enough pocket to make a really good, strong, and specific interaction for a small molecule. And so going forward, that's our next step, is we target something like this, because it has both 2D and 3D information that will make it for a much more specific target in drug discovery. And so with that, of course, I have to thank the National Center for Cryo-EM Access and Training, because they were ever so helpful along the way. They've been amazing. Eugene helped me a ton throughout the process, and we fixed our article today, and we resubmitted the manuscript just this morning, actually, with Eugene as a co-author. And so I know I'm probably leaving some people out here, but I just thank all of them. The guys at Argonne National Laboratory who've been working with us for 3 years now, I think. And then, of course, my lab members at the University of Louisville, and the guys who helped with our computational work. And I'm just leaving you with this meme, but the new meme format for promoter G-quadruplexes going forward. So thank you, and I welcome any questions.
Thank you, Rob. That was absolutely wonderful. I wish you so much luck in convincing people to change cartoons that they're very accustomed to drawing. But I would say that your argument for doing so is very well fleshed out.
Thank you.
Yeah. So we've got one question so far, but do feel free to toss questions in the Q&A if you've got them. There's a question from Lan Guan, who says the class similarity of 0.8 you used is far from the default in CryoSpark ab initio reconstruction. How did you identify that parameter to use?
Yeah, so I guess what I should say is that process of figuring out those parameters specifically for this data set, I mean, it took a solid 8-9 months of just empirically playing around with a few different variables. There's a few different settings that CryoSpark, if you highlight over them, will tell you if it may or may not help when you have poor signal to noise and small particles. But yeah, I mean, it came down to really just playing around with it at every step of the process. In that specific case, yeah, I just did 2D ab initio, 3D at 4, sorry, 2 model ab initio, 3 model 4, etc., with different chosen similarity numbers that I fed in, and just looked at the maps in the end, because in a lot of cases, they just didn't look great, or the resolution wasn't that great, and so trial and error essentially.
All right, thank you. There's a question from Liu Ying Chen, who wants to know how you decided the duplex sequence to use.
Yeah, so it was loosely based off an article, this lab, Salazar, I think, and somewhere in Texas. There was a group that made a duplex quadruplex construct where their insert was a telomere quadruplex motif, and they also used a poly DT region to prevent it collapsing into a duplex. But they used a shorter poly DT region, and they did some chemical biology experiments, but that work kind of stopped. I don't know if it was a decade or more ago. And so, we came across that in the literature, and we thought, well, this is a good starting point to develop our duplex quadruplex system and do our structural studies with it. So it was somewhat based on the sequences they had used in their paper, because they showed that they could get duplex formation without it interfering with their quadruplex, if that makes sense. So essentially, it was just very slightly altered from sequences that somebody else created that were artificial, that showed that it would form a duplex.
Yeah, and actually a related question. Someone says your poly DT chain is pretty artificial. So does the complementary chain interfere with formation of your structure in any way?
Absolutely.
Yeah.
Yeah. So I mean, within minutes, if you mix in, because we've done that and we have SACS data on that and everything. I mean, within minutes, if you mix in the full reverse complement of the G-rich strand, it will convert it to a duplex. Yes. So that's why we have to use a poly DT, or like those early studies did, you would have to do something like in vitro transcription that would sequester the C-rich strand. So, you know, if we were to go back and redo this, or if we were to continue these studies, it's all something else that you could do is potentially scramble the reverse complement sequence just in the region of your loop, but we haven't necessarily made it that far yet.
Yeah, I guess a lot of these questions are sort of dovetailing straight into your answers. So this is great. Keep sending in those fantastic questions. Wei-Jen Tang asks, from your MD simulation, can you now better design your model system to reduce structural heterogeneity and achieve higher resolution? And if so, how might you do that?
Oh, that's a good question. I mean, I don't know. I guess, yeah, we would just have to play around and see. I think that that's a really good point, because if we could, with this starting model, if we could identify a way to modulate or change the poly DT loop region to maybe shorten it, or maybe make it with more specific interactions with the quadruplex or something like that, then yeah, potentially we could make it less flexible. The problem is that, and are my slides still up?
They are, yeah. If you
Okay.
can stop sharing oh yeah,
Well,
I see.
oh,
Yes.
no, yeah, I was gonna
Oh,
say,
you
so
want to share? Got it.
yeah, the problem is that in the region, you know, of the 3' handle here, the region between it and the G-quadruplex, there's essentially going to be a high degree of freedom around the sugar phosphate backbone here. So for reference, to put this into perspective for all the protein people out there, the sugar phosphate backbone is going to have like 6 degrees of freedom. And so this can be a very, very flexible region at this little hinge down here. So we could try to modulate or modify the poly DT loop to increase its stability, to reduce its you know, dynamics, but it's going to remain a case that it will be difficult if one of these is unstacking at either end, because there's so much freedom that could be there that it may be impossible to really reduce it enough to get a higher resolution map. But that would be really cool if we could.
Perhaps the design of a proper drug at the 5' end would actually do that stabilization for you and show that the drug was actually going to perturb the system, or in fact stabilize that to prevent something from happening. Maybe you have to design the magic drug, and once you find that it gives a high resolution structure, that's your validation that, yeah, you did something to the structure.
Yeah, if my mentors are on and listening right now, they're probably sitting there going, that's what we were gonna say. Because you know, they're the drug DNA guys, the gurus. So yeah, that's probably a really good answer. Yeah.
Yeah, all right. There's a couple questions related to MD, and Rob, we lost your video. I don't know if that was intentional or not.
Yeah, let me try to stop sharing and see.
Yeah.
I don't know why it stopped. Yeah, I don't think I clicked it off. Okay, sorry.
Oh, there we go. That's all right. Just, it's nicer to chat when we can see each other.
Yeah.
So there's a question from Eduardo Romero about what molecular dynamics software did you use to run the MD constrained by the EM maps?
We use Amber in all the MD simulations. Okay. And in Amber's manual, there is a little section specifically for EM map refinement that's extremely helpful, and it has actually it has a little example input file that you can actually use, and it explains it really well how to implement that. Yeah.
And were there manipulations you had to do to the map file to use that in Amber?
No, it was
Okay.
surprisingly, I mean, yeah, surprisingly, you just need a .mrc file and it works pretty beautifully right off the bat. Actually, the hard part is trying to figure out, I mean, there's a lot of parameters there that you have to tweak to not essentially get your uh, uh, essentially not get your system to explode by moving so fast into a map. So you have to fine tune some parameters there to get it to slow down a little bit and adjust slowly rather than collapse into it.
And then someone asks, do you think it would be worthwhile to try to go back to the 2 classes you didn't use for your reconstruction? And would you expect those to perhaps be other states in your MD simulation?
Yeah, so right, so I've done the same analysis essentially using the other 2 maps already, and essentially, yeah, they look to be well, so I will say that the MD simulation, starting from the class 1 map that I showed here, I don't know that throughout that simulation it's exactly sampling similar conformations to those other 2 maps, but from our prior and initial MD simulations, we were getting sampled conformations that were very close to those maps as well, but they were distinct from this class 1 map. So yes and no. I have seen sampling in very similar conditions, but I don't think that when starting from the class 1 refinement, because it's seemingly very stable actually in that conformation, it doesn't seem to want to switch back, but that could be a limitation of either A, this could be something that's happening on a millisecond or longer time scale that we can't sample with MD, or B, it could be because of the exact parameters we're using in the force fields or the water molecules or water systems that we're using in the MD simulations that just, you know, that it's just not making it over some energy well to sample those other conformations. But yeah, I mean, we see close, yeah, the sample's close to those other maps.
Yeah, and once you've refined it into, say, the class 1 map, then can you let it free of the map and see whether, starting from there, it would go to one of the other states, or to make one of the other states a target and see if it can go wander over there and find it. That would be kind of forcing it to do that. But yeah, another question about using the cryo-EM map for the to constrain the molecular dynamics. Is it a threshold or envelope kind of restraint that if you allow the threshold to be lower and the envelope to be bigger, that it would actually loop out into other parts of this of the poly T loop and occupy a larger volume just because there's more space for it in the map that you might use. I don't know whether it's a threshold issue, an envelope issue, or whether it just goes by the densities, which vary inside the map all the time.
Oh, that's a good question. You know, honestly, I don't know. As far as the map side, to the former point, we can definitely do nudged elastic bands and those kinds of things to go from one map conformation to another and look at those energies of transitions that way. Yeah, that would be really interesting. But to your latter question, I'm not actually sure how to answer that. You can actually feed in resolution estimates to your map with the restraints of your map, and that will sort of, I guess that will guide the strength of the potential. But I don't know if you can like leave the map, kind of leave it loose to where it's sampling more, because having, I mean, for a poly DT loop that's just in, you know, that's not constrained by bonding or anything in these MD simulations. Number one, we don't we don't really have great parameters for single-stranded DNA and MD simulations that I know of. But number two is that any amount of a potential that you put on this thing, like I was saying before, you know, any amount you can just, they'll crash together. So even with the weakest restraints, which is what I used on the map, the poly DT loop pretty rapidly finds its way into the map and then does not ever, you know, want to leave. But I get what you're saying, but I'm not sure I know how to answer if you can, you know, make the map kind of more expanded to where it's more probable that it'll search more of that space.
Yeah, generally speaking, the lower the resolution, the more concentrated the highest density will be at the center of gravity of the map. I mean, you know, just because, you know, a low resolution map will, in fact, have a smoothly varying function that kind of centers itself in the center of gravity of the map. Some way of getting around that would be to say, okay, inside the envelope of the map, whatever you choose, you have an even distribution of density, which, you know, is really a higher resolution idea once you go to Fourier space, but it actually says within the envelope of the map, if you think that any part of that envelope is okay, then it won't favor that collapsed, you know, go to the center of the map, and it will allow it to wander around inside this plateau of density inside what you've called the envelope. So, I mean, there are just different ways of thinking about what the constraint of a map is. Is it on the highest density, or is it within the envelope at all? And those are two extremes, I guess. It's tricky.
No, that's good. It's good to know.
Yeah, actually, Rob, I have another question because we've been talking a lot about using your maps in the MD, but I wanted to take a step back. You mentioned that you could use a SACS envelope as an ab initio model going into refinement of cryo-EM data, but I think you didn't end up using that, or did you, when you started doing refinement?
No, so the only part of the SACS data that was used in any amount of the refinement was I just used that Dmax estimate, and well, I guess the Dmax, so the length and width roughly of that ab initio, that's what we used for elliptical blob picking, and then from then on out it wasn't used because for a variety of reasons that ab initio reconstruction is very unreliable, and so we don't want to hold too much stock in it in general.
Thanks. All right, I don't see any other questions, and we're actually pretty much right on time. So if there are no more burning questions that people can type as fast as they can, we'll go ahead and wrap up. And thank you very much, Robert. That was really exciting, and I think, you know, often people are pushing for resolution, but this is another great example of, you know, that's not always the way forward in a particular project. So I want to thank our audience for joining us today. We wish all of you well in your cryo-EM endeavors. The National Centers are here to help you push your projects forward, and let us know how we can help. And hopefully we see some of you back, or all of you back, in December for our roundtable on a topic we didn't even touch today, which is particle picking with neural network pickers. So join us again. Thank you, Rob.
Thank you, guys.
Bye-bye.
Bye.